Thanks, everyone. This presentation is titled Modelling Molecular and Immunohistochemistry Tests for Cancer Care, and I'm presenting this on behalf of myself, Dr. Nicola Toda, and the team from the New Zealand Te Aho Te Kahu, which is the Cancer Control Agency in New Zealand. This is the agenda. I'm going to start by giving an overview of the CanShare program. We're then going to look at how genetics test results are used for cancer care. Then we'll look at some modelling examples, and lastly, we'll look at the next steps. The CanShare program is a national initiative developed by Te Aho Te Kahu, the Cancer Control Agency, in collaboration with Te Whara Ora, which is Health New Zealand, the Department of Health in New Zealand. It's a national platform that aims to allow the timely sharing of relevant and accurate cancer data. And it's based on a few guiding key principles, including the fact that it's a clinically led informatics platform that ensures the circulation of data rather than its stagnation. It focuses on providing complete, accurate, timely and shareable data, and it's standards-based. And the standards it's based on are SNOMED CT, FIRE and the HISO data standards in New Zealand. So this diagram provides a high-level overview of the program, highlighting the key participants in the data sharing, including structured pathology, radiation oncology, systemic therapy, surgical reporting, multidisciplinary meetings, and cancer registries. The primary aim of CanShare is to support clinicians in delivering the best possible cancer care to its patients. However, the outcomes of the work also include advanced analytics capabilities for monitoring cancer care across the country. This slide shows some of the different types of cancer data that are shared between the participants you saw previously. But today I'm going to particularly focus on the pathology reports, and particularly the immunohistochemistry and the molecular biomarkers that are used by the downstream activities for diagnoses, staging, prognosis and treatment. So why are these tests important to us? Well, cancers develop when something goes wrong with the genetic structures in a cell. And by being able to access and understand the genetic test results, we can assess the risk of cancer and guide preventative measures. We can help with the staging and the prognostic scoring, and it can help to guide the treatment choices by predicting how a patient with certain genetic characteristics is going to respond to those treatments. And there are two main types of tests we're going to talk about today. The first is the immunohistochemistry test, which looks for specific proteins in a tissue sample using antibodies that bind to the target protein. And the second type of test we'll look at are molecular tests, which analyse the molecules like DNA and RNA to directly detect genetic changes. So here is an example of a range of test results that you might see on a lung cancer histopathology report. This one's from the International Collaboration on Cancer Reporting, or ICCR, and it's their ICCR lung cancer protocol. As you can see, not sure if I've got a laser, oh I do. As you can see, it's got a bunch of molecular tests that look for variants in a range of relevant genes, including EGFR, ALK, ROS1, BRAF, MET, and HER2. And it also has an immunohistochemistry result down here, which looks at the PD-L1 protein. Here's another example for invasive carcinoma of breast, which includes two immunohistochemistry tests, one that looks at the estrogen receptor, and the other looks at the progesterone receptor, and then a third test result that includes both immunohistochemistry for the HER2 status and the second test, which is by in situ hybridization, which is a type of molecular test. And you might notice that the in situ hybridization has results like positive or amplified and negative or not amplified, and then the immunohistochemistry has test results like scores like 0, 1+, 2+, 3+, and also interpretations like positive, negative, and equivocal. As I said earlier, these test results are used for a range of purposes, including downstream staging. So here is an extract from the HACC Staging Manual, Chapter 48 for breast cancer, and it shows how these statuses, particularly the ER/PR and HER2 statuses, are used to determine the clinical prognostic stage group for the cancer. And you'll notice that when you get to staging, all the staging tables care about is whether those statuses are positive or negative. It's very high level. Another reason to do these genetic tests is to predict the effectiveness of certain treatments. So here's an example of a New Zealand systemic therapy regimen, which contains trastuzumab. And trastuzumab specifically works to treat cancers that produce too much HER2 protein. So it's only effective on HER2 positive cancer patients. And you can see in SNOMED CT that trastuzumab, the substance, is a child of human epidermal growth factor receptor 2 inhibitor, or HER2 inhibitor, and that's why it's primarily used against HER2 positive cancers. In representing these tests using SNOMED CT, we've followed these modelling principles. Firstly, we only model concepts for which there's a clear use case, and in particular, we focus on those test results which are either used in a pathology report or for staging, or they're used to determine the eligibility for a systemic therapy. We also make sure that we use a realistic and manageable level of detail to satisfy these use cases, and we need to ensure that the concepts remain stable and relevant across time, particularly with the ever-changing genetics knowledge and different molecular tests that become available. We've also made the decision to use observable entities for the tests because of its rich concept model, and clinical findings for the results, because that means that in your reports, even if you just have the result, you can trace it back to the observable entity and get that rich definition with all the information from there. So far we've looked at both direct tumour testing, indirect or circulating tumour testing, and non-tumour or germline testing. But today we're going to focus on these tests. So we have immunohistochemistry tests that are just focused on the protein that it's looking at. But we also have immunohistochemistry tests that are surrogates or replacements for a molecular test, and we'll talk more about those soon. And lastly, the molecular tests, which look at a single variant in a gene. In modelling these tests, we're using the observable entity concept model with the component attributes set to a descendant of protein, and they're typically oncogene proteins, or a descendant of cellular and or subcellular abnormality. And here we have the genetic alterations, which are morphologic abnormalities, and you'll see things like gene amplifications and deletions and rearrangements. The property attribute is either set to presence or substance amount. The scale type is always set to ordinal value. Adheres in is either neoplasm for proteins or a subtype of subcellular structure, such as the specific gene or chromosome. And if the observation adheres in a subcellular structure, then the inherent location is a neoplasm. The time aspect is always a single point in time, and the technique, if it's included, is either molecular or immunohistochemistry technique. So this is the model of the observable entity concepts that we use to meet these use cases. Firstly, we create a technique-free observable at the top, and this represents a particular molecular status with no technique, and we use this concept for some of our downstream use cases like the staging, treatment and biomarkers, where the technique is not as important as the actual interpretation or result. We then create two technique-specific observables for the molecular test, and if there is a surrogate immunohistochemistry test, we create one of those, and we create concepts for our protein-focused immunohistochemistry tests, which represent the protein status. So, as I said, the technique-free molecular status concept is used for downstream staging, treatment and biomarkers, and the other three concepts that have a technique are typically used on the pathology reports. So we're going to take a look at the templates for each of these concepts. So template 1, this models the technique-free concept at the top, which has a molecular intent. The purpose of the test is to determine whether or not a particular gene alteration is present. And we've created a few concepts that are groupers, like this one, the HER2 alteration status, with a component of genetic alteration, and then some more specific observables like HER2 amplification status, where the component becomes more specific down to gene alteration. Template 2 then models the observations that are performed using a molecular test, and their definition is essentially the same as their parent, but just with the addition of the technique of molecular test. And then when we come to representing those immunohistochemistry tests, where the protein that's being observed is a known surrogate or replacement for a specific cancer-related genetic alteration, immunohistochemistry tests are often used first because they're cheaper and they're faster than the molecular test, and the genetic alterations often lead to a specific change to the protein expression that can be measured and serve as a direct indicator for the underlying genetic event. So where there's a one-to-one association between the protein expression and the genetic alteration that causes it, these tests can be defined either in terms of the protein that's being observed. Down here it's the receptor tyrosine protein kinase ERB2, or the molecular alteration that is a direct indicator of. In this case, it's the presence of gene amplification in the ERB2 gene structure. And those axioms are all necessarily true, and then the protein observation is also sufficient to define this concept. So we've got two axioms. One of them's sufficient and the other is not. We've got a defined axiom and a primitive axiom. And interesting animation, but for our immunohistochemistry tests, the ones like PD-L1 or ER, PR status, where it's just the protein that is being observed and it's not used as a direct indicator of a molecular test, we represent these protein statuses using an immunohistochemistry observable like this, where the attributes are as I explained before. The component is the protein, so this is the one for PD-L1, and the technique is the immunohistochemistry technique. So the modelling templates that I showed you are based on the stated definition. That's what the author creates. And then once these are classified, they get placed into a hierarchy like this, with the technique-free observable at the top, which, as I said, is used for staging and downstream treatment purposes. This is the presence of amplification of the ERB2 gene. And then we have the immunohistochemistry observable, which looks at the protein and the molecular test underneath as two different types or two different ways of finding out the HER2 status or the ERBB2 amplification. So we've covered the first four observable entity templates. Now let's have a quick look at the other side. This is the test results or the clinical findings. So we're going to particularly look at these two bottom classes, which are the clinical findings for the molecular test and the immunohistochemistry test, and we'll come back to the HER2 status. And we'll look at the results of the immunohistochemistry test. As we saw before, they had a score of 0, 1, +2, +3+, and the molecular tests, which have the result of the detected or not detected in terms of the gene amplification. So when we're modelling these clinical findings, we set the interprets attribute to the observable entity that we created before. So we're linking back to that rich concept definition that we had. It interprets the presence of the amplification of the B2 gene, and the interpretation is the result. So it's the 3+ immunohistochemistry score. And then for the molecular test results, once again, we have the interprets, which links back to the observable entity with all that rich definition that we've created, looking at the presence of the amplification of the OB2 gene by molecular genetics technique, and the interpretation then is the detected. So in terms of next steps, the New Zealand team is working with Scott Campbell and the pathology working group towards agreement on the details of these templates. Once the templates have been agreed on, New Zealand will then promote all of their observable entities and clinical findings that could form to those templates to the international edition so that they can be shared with the community, and we encourage other countries to contribute any additional observables and findings content that they have so that we can share them all with each other. And New Zealand will continue to develop techniques for new types of molecular and immunohistochemistry tests, as I said, focusing on the ones that are relevant to the structural pathology reports for treatment, eligibility, and staging purposes. And for those of you who weren't in the last session, just a quick plug for my book, The Essential Guide for SNOMED CT, which was published in August. It has 33 chapters, starting from what SNOMED CT is and why it's important, through to advanced techniques for using SNOMED CT with FHIR terminology services. And there's the discount code again, and the link to get there. Thank you very much, everyone.
Any questions from anyone in the room? Here we go.
I'm not sure if we have Nicola on the line. She was going to be.
Darren was going to check on that, I think.
Is Nicola Toder on the line? She was going to join us.
I'm here, Linda.
Oh, wonderful. Thanks, Nicola.
I have one question in the room so far.
Thank you very much. Thomas Debertshäuser from Charité. I've been struggling with similar problems over the last year, coming up with some systematic. I really like the approach you've taken. I'm wondering, could you go back to template number 3?
Yeah, that was the really interesting one where it has the 2 axioms.
Yeah, because I think in this use case it is well known and well established that
That's Nicola. Meet Nicola Toder, everyone.
Okay, I was referring to the component of gene amplification because
Can you get the presentation back, please? As lovely as it is to see Nicola, it would be nice. Thank you.
Right, because the usual pathomechanism in R2 is that there is a gene amplification and therefore there's more protein and therefore you see more protein.
Yes.
But if you work in precision medicine and with rare cancers, that's not necessarily always the case. There might be other mechanisms that lead to more protein, which means more transcription or a change in fusion, regulatory, whatever. So I would be careful to make this this axiom fixed, that the component always has this gene amplification as a cause. That might be true in cases where it's well established, and I give you 100% it is well established in breast cancer, but it might not be true in rarer cancer forms.
Yeah, that's a really good point. Thank you for raising that. These concepts specifically refer to an observation where you are looking for the genetic alteration using the protein. We certainly try to only define them where there is an established surrogacy, but obviously, as you say, there are other mechanisms that can cause it. So I wonder if Nicola has anything to add to that?
I don't think I can add more. You've explained it well from our point of view, Linda.
Okay, thank you. John?
Thanks, Linda. This was a great presentation. Going forward to where you represented the findings as the response values, the interpret's attribute is implying a binding to the observable.
Yeah.
Have you found a need to associate a binding strength so that you can lock in that answer list as being the only answer list that's applicable to that observable? And if so, is there a way that you can see that being represented in SNOMED?
Well, binding strengths are specifically in relation to terminology binding to an information model. So in respect to the logical information models that the New Zealand team is developing, that's where their bindings to these observables and the clinical findings are. They're currently not using a binding strength because they're using those bindings to auto-generate the FIRE questionnaire forms directly from those, but that's an interesting proposal. Do you have anything to add to that, Nicola?
I don't think so. Thanks, Linda.
Okay, thanks.
Hi, so I have sort of a follow-up on that, because what happens if you use like the naked only like qualifier value detected as an answer?
Yeah.
Would that be doable, or would that ruin everything?
And that's something that's often proposed. And that works well where you're using FIRE and you've always got the observable clinical finding pair. And if you're always keeping the observable clinical finding pair together, then you've got the rich definition of the observable and everything about it in terms of what it's looking for, the gene, what gene alteration. But by using the clinical finding, you get the observable for free and you only need to store the one clinical finding code. So for implementations that may not be quite so advanced, that are just recording the clinical finding, it's sometimes hard to keep that pair together. So it's a way of ensuring that your result always knows what observable it came from.
I realize
I
this,
can
it's just you will end up with a lot of clinical findings for every observable, and in this case there's only like detected, not detected, but eventually you will have all the entire scale from a little present to, and then for every observable there's a combinatorial explosion potential here.
Yeah, and one of the principles that we talked about earlier was the scalability of that, which is why we're specifically focusing on those results that we require in our structural pathology reports. But Nicola, I heard you wanted to say
Yeah,
something?
just to add to that, for one of our implementations, it is a anti-cancer medicine eligibility, anti-cancer medicine prescribing system. And so in that system, the fields that are being filled out would be more general than the exact observable that is used as the interpret's value for the findings. So we can have a more generic, broader HER2 amplification field, and it can be populated with any of the findings which come from a pathology report, and each interpret different observables.
We can talk afterwards as well, yeah.
Yeah,
Yes.
me again. It just came to my mind. I think there is in some newer guidelines the clinical finding of HER2 low. I'm not sure whether that's already in your national guidelines. I think it might be European. I'm not entirely sure. I'm just wondering because this is a clinical finding that is dependent on a immunohistochemical finding of 1+, plus additional immunohybridization results of at least 2 times, 5 times fold amplification or whatever, which means that would be dependent on 2 parameters. Are you able to represent that within your systematic?
The 2 parameters of the score plus the
clinical finding,
interpretation.
the clinical finding being an interpretation of 2 observable entities.
clinical finding. I'm not sure if this answers your question, but let me try. We use the information model to separate the score, like 0 plus 1 plus 2 plus 3 plus, from the interpretation of, low, positive, high. And you alluded to the fact that these interpretations are changing in the ICCR protocols. And that's something that Nicola and I discussed beforehand. So you obviously know your stuff. But yeah, we use the information model to separate them, and we don't attempt to link them other than to say that this is the score and this is the interpretation. Did you want to add to that, Nicola, and try to target
Yeah,
things?
I think if I understand the question, we something that we're currently looking at modeling these newer HER2 low, ultra low, etc., because they've just coming into New Zealand standards. With our template within the, we could have two different observables that are being interpreted using using multiple role groups, I believe.
Thank you.
Yes, good point, Nicola. Thanks for that.
Oh, one more? You okay with one more?
Thank you. I think this is a very basic question, but I was wondering in the modeling, why didn't you add a finding site like we found it in this gene?
Because finding sites used in the clinical finding hierarchy, we were...
Yeah, it's for model 4,
Oh,
with the
for the
clinical
result?
findings. Yeah, yes,
Oh, for the
indeed.
result, I see. Because that information was captured in the observable entity in terms of the here's in and site of the observable, so this interpretation pair is sufficient because that information is captured inside the observable entity.
But like it's really simplistic that I'm asking this, but with a fracture of the arm, and you say we did it with radiology, we interpret the X-ray and we say it's a fractured arm, you will still find in the modeling also a finding site of the arm. So I don't see this difference with genes then, but that's my feeling with this.
Yeah, well, thanks for that feedback. We chose not to do that. Thanks for the feedback.
Thank you, Linda. I think, Nicola, that concludes our sessions for today. It's lunchtime, so thank you, everybody. Thank you for attending, and enjoy the afternoon.